Hi, in this video we're going to discuss an important aspect of vision. But before moving on to the topic proper, can you guess what you're seeing on the screen? So some of you might be saying that it's a blue car, or if some may know the more details, you might be saying that it's a blue Rolls Royce. But how did you understand that? Was it written on the screen? No, right? But when this image was shown onto your retina, your brain could process it as a blue car or even a blue Rolls Royce. So how does the brain do that? Now I'll just demonstrate a few attributes of this image. See, suppose I'm going to increase or decrease the brightness of the image. Are you able to see it? No, right? Now here we can see that there should be proper lighting of that image for it to be visible for us. So that is meant by light sense, or that is how the eyes has got the ability to adjust the light so that a clear image can be formed on the retina as well as the cortex. Now I'm just going to adjust its contrast. You can see that when the contrast is decreased or as well as increased, the car will not be visible. See, visible properly, you can't exactly tell the details of the car, right? Which means a proper contrast sense must be present in order to perceive the contrast correctly. Now I'm going to change the saturation of the colors. You can see that of course your brain should be able to recognize the color blue to see it as a blue car. And now I'm going to change its sharpness, and you can see that the details of the car are no longer visible, which means the form of the car should also form sense should be there in order to see this car properly. So all these points to the points that all these components should be correct for the proper perception of the image by our brain. So on that note, the topic for today's presentation is visual perception. So in this presentation, I'm going to tell about the history, some experimental evidence, the definition, the basic steps of phototransduction, then about light sense, form sense, contrast sense, color sense, depth perception, as well as some applied aspects. So first let's see the history. So two scientists named Thorsten Niels Wiesel and David Hunter Hubel, they got Nobel Prize in Physiology in 1981 for their information processing for discovering the information processing in the visual system. Okay, so it was Hunter Hubel as well as Thorsten Wiesel. So now moving on to the experimental evidence. So how do the scientists find out that there are specified cells present in the cortex to perceive an image? So for that, they inserted a microelectrode onto the primary visual cortex of an anesthetized cat in such a way that this microelectrode will record the response to a stimulus, and the stimulus here was a bar of light. So basically they showed a bar of light onto the receptive field of the anesthetized cat and recorded the firing. So they found out that whenever the bar of light was shown at a particular orientation, there was increased firing of the neuron. So they marked that area as crosses, and then they further changed the orientation and position of the bar of light and to see whether there was this response to the bar of light. So in the area adjacent to this, they found that when the light was off, the action potential was recorded. So they marked that area in, to show that the response was when the light was off. Okay, so they found out that this is a receptive field of that particular neuron into which the microelectrode was inserted. Next, they changed the orientation of this bar of light to see whether there was any effect, but they found out that there was no effect whatsoever. See, this is they are just marking the areas where there is response, and now they'll change the orientation. See, they've changed the orientation, but they could not record any action potential onto the neuron. See, now they are moving it to the vertical orientation. So see, they couldn't record anything unless the orientation was this particular orientation. So that is how they found out that there are cells in the cortex that respond to a particular orientation of the image that is shown. Now the definition. So visual perception is a complex integration of light sense, form sense, sense of contrast, and color sense. Now, we should first know what happens when light falls onto the eyes. So we know that light energy is converted to neural signals by the visual pigments present in the photoreceptors. And which are our photoreceptors? We've got rods and cones which has got discs present on them in which we've got the visual pigment. Now what happens when light falls onto the visual pigment of these rods and cones? See, suppose this is a rod and this is the disc of that particular rod. So when light falls onto the visual pigment present, which is rhodopsin, it in turn activates G protein transduction, which in turn will activate cGMP phosphodiesterase. So what will happen if phosphodiesterase is activated? See, actually there are some cGMP gated sodium channels present on the rod which allows continuous entry of sodium into the cell, right? So this will open only if cGMP is present. But when this phosphodiesterase is activated, it will convert this cGMP to 5-GMP, which means there will not be enough cGMP to open the sodium channel. So in the dark, there is continuous flow of sodium into the cell, but in the light, there is no entry of sodium, which means the cell will be hyperpolarized. So see, in this image, you can see that there is the sodium current present when the photoreceptor is in the dark. But when the photoreceptor is in the light, there is no sodium current and this new neurotransmitter is released. Now what happens when there is release of neurotransmitter? See, in the dark, I said there is a release of neurotransmitter, which in turn will activate this inhibitory bipolar cell and no neurotransmitter will be released from the bipolar cell. But in the light, the photoreceptor is not releasing any neurotransmitter, but this will activate the bipolar cell and in turn release neurotransmitter, which will activate the ganglion cell. So remember, in the dark, the ganglion cell is not activated, but in the light, the ganglion cell is activated. So, if an image is shown on is present, that particular object will give off light rays to our eyes, which converge with the help of a lens to form an inverted image on the retina. So see, this is the upright image, but the image that is formed on the retina is inverted. Okay, so this is detected by ganglion cells, and the axons of the ganglion cell form what is known as the optic nerve. So we should know the visual pathway here. So the ganglion cells, axons form the optic nerve, which will cross over at the optic chiasm. See, the nasal fibers cross over, whereas the temporal fibers will just move as such, and they form what is known as the optic tracts after the optic chiasm. After the optic tract, they reach the lateral geniculate nucleus. So lateral geniculate nucleus has got 6 layers, of which the first and second layer is involved in the magnocellular pathway and the rest 4 is involved in the parvocellular pathway. And via this pathway, they reach the primary visual cortex, which is area 17, also called V1. Okay. So once these signals reach the cortex, we should see what happens. We said that it reaches the area V1 of the cortex. Now, just like in any other areas of the cerebral cortex, it's got 6 layers. So these impulses which reach from the lateral geniculate nucleus, they reach the area 4C alpha of the cerebral cortex, and from there they give off with projections to the other layers of the cortex, right? So from the area V1, we've got 2 pathways. The first one is called the dorsal or the parietal pathway, which is primarily concerned with motion, form and 3D position. See, from here you can see that from this area 17, we've got this dorsal pathway, which is concerned with motion, the form, 3D position as well as motion. Next, we've got a ventral or the temporal pathway, which is concerned with the shape and recognition of form and faces, which means it is more concerned with the visual detail and color. So from the eyes, via the optic nerve, it reaches the lateral geniculate body, from there to the primary visual cortex, and from there to other areas by this dorsal as well as ventral pathway. So why should impulses move to other parts of the cortex other than V1? See, Here you can see that there are different areas on the brain which are responsible for vision. And here we've got the function of each. See, for example, area V8 is related to color vision. So from V1 it moves on to area V8 for proper processing of the color. Similarly, for example, see, V3A is responsible for motion. So see, from V1 it moves on to V3A for proper detection of motion. So like that, each of the secondary visual areas have got a function. So from the primary visual cortex, they move to the secondary visual areas for further processing, right? So now we'll move on to visual perception. So the first one is light sense. So what is meant by light sense? It is the awareness of light. Okay. So our eyes have got the ability to adjust the amount of light that should reach the retina as well as the cortex. How? So two basic mechanisms that demonstrate this is a visual adaptation. So there are two types of visual adaptation, that is dark adaptation as well as light adaptation. So what is meant by dark adaptation? So dark adaptation is a process that refers to the gradual increase in light sensitivity of the retina when in low light conditions. What exactly does that mean? See, in your practical lives, you might have experienced that when you suddenly enter a room that is dark, you'll not be able to see immediately, but after a few minutes you'll be able to see that properly, right? So that is because of dark adaptation. Your eyes will adapt to that darkness. And what is the mechanism? First mechanism is by regeneration of the visual pigment. All your visual pigment that have been will be suddenly be cut off when you enter a dark room, right? So even though it takes some time, there will be regeneration of the visual pigment, which will allow clarity of vision. Next, there will be an increased pupillary diameter so that more light can enter the eyes so that you will be able to see properly. And also there will be increased retinal sensitivity, which means the retinal sensitivity increase so much that even a photon will be able to activate a photoreceptor. So here you can draw a graph. So you can see a graph between retinal sensitivity and the minutes in the dark. You can see that the cone adaptation occurs much earlier than broad adaptation. But have you ever experienced that you can see color better in a dark room? No, right? So what does this mean? See, even though cones adapt better, their sensitivity is less. So in a dark room, because the room is less illuminated, the cones will not be able to detect its color. But whereas rods, even though they adapt slowly, their sensitivity is very high. So when a single photon will be able to activate that rod so that we'll be able to see the object even though we might not be able to decipher the colors. Okay. So that is meant by dark adaptation. Now what about light adaptation? The process by which the retina adapts itself to bright light is called light adaptation. So see, in here also in your practical lives you must have felt that when you suddenly enter a bright lit area, you will not be able to see clearly for a few minutes. But after that you will be able to adjust to its bright light. So that is meant by light adaptation. So here also the mechanism is first of all neural adjustment, visual pigment mechanism and a decreased pupillary diameter. So what is meant by neural adjustment? See, even before you enter a brightly lit room, your cortex will be able to anticipate that the light entry is going to be high and there will not be sufficient pigment enough for you to see the objects clearly. So that is the anticipatory effect which occurs even before you enter the room. And once you enter it, you've got this transient effect which is due to neural adjustment. The retinal sensitivity will decrease and the neurons will adjust to the increased bright light. And then we've got this photochemical effect which is due to this visual pigment mechanism. Basically, it's just the opposite of what happens in a dark adaptation. And there will be a decreased pupillary diameter so that less light will enter the eyes. Okay. So this, this is an image. You might perceive this as an image of a girl who's dressed up and it's an angel, but she can actually appear like an angel if your light sense is not proper. Okay. Now, next is form sense. So form sense is the ability to discriminate between the shape of objects. So a measure of form sense is visual acuity, and mainly it's a function of fovea. What is meant by fovea? See, fovea is this region, this depression on the retina wherein you've got a huge increased concentration of cones which will allow proper vision of proper understanding of the details of an object. So only if an object, the light from an object falls onto the fovea of our eye, that we'll be able to correctly identify the details, right? So why is fovea so important, or how is the image formed on the fovea of the retina so clear? That is because in the fovea, first of all, the cones are densely packed and each synapse onto a single bipolar cell. So see here, this part shows the region which is the foveal region, whereas this is the peripheral region. You can see that each photoreceptor synapses onto a single bipolar neuron, which in turn synapses onto a single ganglion cell, right? Whereas in the peripheral region, you can see that multiple photoreceptors synapse onto multiple bipolar cells, which in turn synapse onto multiple ganglion cells. So, because we've got such a direct pathway, there is increased transmission of impulses from this photoreceptor to the brain. Moreover, there are very few overlying cells and no blood vessels, which so the details are correctly transmitted faster into the brain. And in the brain, the cortex also, this foveal region or the macular region has got a huge representation. We can see that this whole area is just for processing the image that is formed on the fovea or the macula of the retina, right? So because of this large representation, you can clearly see the details of the image when you look at it. Okay. So which is a pathway that is responsible for clear vision? I said that that's a huge, in the primary visual cortex itself, that we've got a huge representation for macula, and for further processing we've got a specialized pathway which is a dorsal pathway. See, remember we had said that there's a dorsal pathway which is concerned with the form, 3D position and motion. So how does that pathway move? So from the primary visual cortex, signals flow to the posterior mid-temporal area and upward into the broad occipito-parietal cortex. Okay, and from there it interacts with the signals that are formed at the somatosensory cortex, and like that they'll be able to analyze the three-dimensional aspects of somatosensory signals. Okay, so that is about form sense. So now here you can see that this is a picture of a beautiful girl, but if your form sense is not proper, you might perceive this like this. So that is how important our form sense is for proper visualization and of details of the object. Next we'll see about the contrast sense. So contrast sense, so suppose this is the image that is being projected. In the cortex, there will be maximum activity at the edges, which means our cortex is designed in such a way that it responds maximum to the edges of the image. So this encoding actually begins at the ganglion cell level itself. Remember, after the photoreceptors, we've got the bipolar cells and then we've got the ganglion cells, right? So the contrast is encoded right at the ganglion cell level. So this ability to perceive sharp outline is due to these specialized ganglion cells. So this is an example of a receptive field of an on-center ganglion cell, and this is the receptive field of an off-center ganglion cell. So let's see more about it. See, in an on-center ganglion cell, if light falls onto the center of the ganglion cell, there is increased activity for the ganglion cell. So that is why it is called on-center ganglion cell. That is, if the light falls on the center, it will be on. Whereas for off-center ganglion cell, if the light falls at the center, there will not be any activity. So this is why it is called off-center. So when light falls on the center, the cell will go off. See? Next, what if the light falls on the periphery? See, if the light falls on the periphery of this on-center ganglion cell, the cell will be off. That's why it is called off surround. Whereas if the light falls on the periphery of this off-center cell, this cell will be on. That is why it is called on surround. So we've got two types of ganglion cells, on center as well as off center. So this is another image which shows the same principle. See, suppose this is the receptive field and we've got light at the center, and you can see that there is impulse transmission in the on center ganglion cell, right? But here the light is shown onto the periphery and not in the center, and you can see that there is impulse transmission in the off center ganglion cell. So this is how ganglion cell encodes the contrast. So at the border between light and dark areas of the image, the neighboring cells respond in opposite way. So that is why these edges are so clear to us, because one cell will accentuate it while other cell will decrease it, so that the edge will be clearly visible. Now, visual cortex detects not only the existence of lines or borders in the different areas, but also the direction of orientation of each line or border. So how is the orientation being detected by the visual cortex? That is because in the cortex we've got orientation columns. So the visual cortex are arranged in vertical columns called orientation columns, which help us to understand the orientation of each of the image that is being formed. Moreover, we've got cells, different types of cells present in the cortex, like simple cells, complex cells, as well as hypercomplex cells. So simple cells respond to the orientation or bar of light that got a particular orientation, whereas complex cells respond to a linear stimulus which is moved laterally without the change in its orientation, and hypercomplex cells process it further. Okay, so it is because of these cells that the orientation, that means each image is being processed in detail. Say for example, if when I show this image to the retina, an inverted image will be formed on the retina as well as the lateral geniculate body. Okay, and this in turn by means of optical pathway will reach the cortex and will be processed by a simple cells. So see, you can see that the simple cells are more concerned with the orientation of these lines, right? And the complex cells have got a combination of receptive field of these simple cells, so it will detect these lines. And in hypercomplex, they will further process these lines, and this in turn is encoded onto this orientation columns. So by the time it reaches this is processed, the primary visual cortex will be able to identify it as an image like this. But only if it is projected onto the secondary association cortex, that they will identify that it is an alphabet A, right? So this is how a processing takes place inside our cortex. So now you can see that this is a beautiful picture of Mona Lisa, but if your contrast sense is not proper, you will just be able to see like this, wherein the details are not visible properly. Next, we'll see about color sense. So color sense is actually encoded at the level of the retina itself, because we've got three types of cones. One is the L cone, which is responsible for transmitting the longer wavelength. Then we've got the S cone, which is more sensitive to the smaller wavelength. And then we've got the M cone, which is more sensitive to the middle wave. So, and even at the ganglion level also, there is color coding present, and all these impulses, see, so from the cone cells, and it moves on to the P ganglion cells, and from the P ganglion cells via the optic tract as well as, or via optic nerve as well as optic tract, it reaches the parvocellular layer of the lateral geniculate body. Remember, the last four is the parvocellular layer. So from there, via the parvocellular pathway, it reaches the primary visual cortex, which is area 17. Now once it reaches the primary visual cortex, here we see some details. So as I said before, the cortex have got 6 layers, and to understand color properly, we've got specialized color blobs present in our cortex. So the impulses that arise from the lateral geniculate body via the parvocellular pathway, they reach the layer 4A as well as 4C beta of this particular cortex, and from there it gives off its projections to these color blocks for further processing, and from this primary visual cortex it gets projected onto the area V8 for further processing of the color vision. So this is how color is encoded in our cortex. So it is because we've got a proper color sense that this image is so colorful, otherwise it would have been something like this, in which you will not be able to perceive the colors properly, even if your cones are, all the 3 cones are present. Now, an image will not be complete without depth perception. So because we've got a binocular vision, because we've got 2 eyes, and because it's got its angle of vision coincides, we can perceive the depth of the objects also. That means we can have this 3D image. So depth perception is the determination of the distance of an object from the eye, and it is mainly by three mechanisms. One, by knowing the sizes of the image of known objects onto a retina. So suppose you see a pen in front of you. You know the pen is small enough, so it must be only at this much distance, right? So similarly, if you know the size, roughly the size of an object, your brain automatically recognizes how far it is from the body. Next is phenomenon of moving parallax. See, when you turn your head, the objects that are nearer to you will also move, right? So if you move to the you can feel that your visual field is also moving, but objects that are far away will not move. So this is known as phenomenon of moving parallax. And finally, the phenomenon of stereopsis or binocular vision. This is very important for depth perception. It is because we've got a normal binocular vision that we are able to easily climb down the stairs, because those who have a monocular vision will not be able to perceive the depth of each stair, and it will be very difficult for them to perceive it. Okay. So see, this is an image which shows how our eyes perceive the depth of an object. So as I said before, if the size of the object is already known, the brain will process it to know how far it is from us. And if it is an unknown object, by the process of stereopsis, it will be able to understand how far the object is. Right? So that is about depth perception. So this beautiful picture of Elsa appears like this because her depth perception is intact. Otherwise, it would have appeared something like this. Okay. Now moving on to some applied aspects. What will happen if our cortex are not able to process all these information? What will happen if there are cortical lesions? See, we've got a condition called visual agnosia, in which the patient cannot understand the meaning of what he sees. So suppose you show an apple to him, he'll just describe it as a red and round thing, but will not recognize it as a fruit that we can eat. Similarly, visual verbal agnosia. They'll be able to see the words, but they'll not be able to understand what it says. That is meant by visual verbal agnosia. And prosopagnosia is a failure to recognize familiar faces. Now, all these occur when there are lesions in the dominant occipital cortex and adjacent temporal and parietal cortex. Next, if you've got diseases in the temporal lobes, like temporal lobe seizures, there will be distortion of the images. Like, for example, they seem to recede into a distance, which is called pteropsia, or they may appear too small, as in micropsia, or they might seem too large, as in macropsia. And with parietal lobe lesions, the object may appear askew or even turned upside down. Now, if there are lesions in the vestibular nucleus, you can have an illusion that the objects are tilted or even that the straight lines are curved. Now, that is due to a mismatch between the visual image as well as the vestibular input into the visual system. So, this just shows how important the vestibular input to the visual system is, because otherwise you feel that the objects are tilted. Now, can you guess which movie is this particular video clip from? I think at least some of you might have recognized this as Alice in Wonderland. So this was a favorite movie of kids when they were small, in which this girl feels that the object is small, or after some time she drinks a potion and the objects are huge. Well, actually there's a syndrome called Alice in Wonderland syndrome, in which it's a perceptual disorder which is characterized by distortions of visual perception, body schema, and experience of time. So, in this particular disorder, your perception of the outside world will be completely different, at least transiently. So, the causes of Alice in Wonderland syndrome are head trauma, epilepsy, Epstein-Barr virus infections, or even brain tumors. And in Alice in Wonderland syndrome, there can be a series of symptoms, even if it is transient or not. But a huge variety of disorders can take place, like chloropsia, green vision, synopsia, blue vision. You've got a huge list of symptoms that can occur due to a disorder in visual perception. So this just shows how important proper processing of the visual image in the cortex is necessary for to understand the objects normally, right? So to summarize, today we've discussed about in the introduction we've talked about the history, the experimental evidence, then we've seen about the photo transduction, the basic steps, then about the light sense, the form sense, the contrast sense, color sense, and also some applied aspects. So these are my references, and I hope you've understood how, at least got an outline of how the cortex processes the different attributes of an image. Thank you.